Hello, biology. This is 24.1, and we're going to talk about DNA. All right, we're going to talk about genetics and how all that works. Now we'll talk about down to the very smallest of things in the cell, which would be considered DNA. And of course, the work of Mendel, the father of genetics, and many others kind of led to them discovering deoxyribonucleic acid, which is DNA. You can see that on page 487. But it is a nucleic acid, so when you hear nuclear, sometimes they're talking about at the cell level there. James Watson, Francis Crick, these are guys who actually came up with the structure of DNA, which is what we're going to talk about a little bit today. And they also revealed, their discoveries revealed that each gene of an organism is part of the DNA molecule, and each gene would control one particular trait. And it's kind of all encoded into that DNA molecule. Now, we've talked about, just give me a second and I'll let you do that. We've talked about each cell has 46 different chromosomes in the nucleus, and then every somatic cell in your body has 46 molecules of DNA, is what that means. So it's pretty cool to read down the bottom of 47, top 488, but I do want to kind of go over it. If you were to have the information from your DNA in from one cell, written on books, they say it would take 400,000 pages at about 500 words per page. And then it says each cell of your body would basically be able to contain the information to fill a library of about 1,000 books. So yet it only takes up about 0.0007% of your body weight. So that's pretty crazy. So between 60 to 100 trillion copies of genetic information in your body, because that's how many cells you have. All right, so short segments of each DNA molecule are kind of coiled onto protein spools called histones, and we'll talk about different types of histones. But chromatin is loosely coiled DNA. All right, so it's what's happening under normal processes of the cell. It's called chromatin. Now, when it's getting ready to undergo cell division at the nuclear level, then they bundle really tightly together, and then they call that the chromosome. So basically, there's like the same thing, but it depends on the packaging. So loosely, it's chromatin, tightly bundled chromosome. So you understand that there. All right, so what is the structure of DNA? You definitely want to follow along, look at 489 while I'm talking about it. So besides DNA, there's another special type of nucleic acid called ribonucleic acid, or we call it RNA. And each DNA and RNA, they're composed of nucleotides, which is just the molecules that make up the DNA and RNA. Each nucleotide is made up of 3 different parts. All right, so this is where I want you to look at the double helix structure of DNA. All right, so you see the legs that go down, what that looks like a ladder there. That's basically the backbone of the DNA, and that is consists of the nucleotide chains there. Okay, you've got those 3 main parts. Let me go back to the 3 main parts there. The sugar molecule, which that's the deoxyribose in DNA, ribose in RNA. That's the sugar molecule. And then you have a phosphate group, and then you have a nitrogen-containing base, which we'll talk about in a minute. So those 2 long nucleotide chains there, that it's kind of looks like a ladder that goes down through there. The legs of the ladder, those are going to be the nucleotide chains, and they're going to be connected by the bonds between the sugar and the phosphate molecules. The rungs of the ladder, the parts that go like this, those connect the legs of the ladder. Those are going to be the nitrogen containing bases. All right, so the 2 legs, the phosphate group, the sugar, connected by the bases, which there's 4 different types of bases, and we call that base pairing, and that means that only certain ones go with certain ones. That DNA structure is taken and then it's twisted, and it's called a double helix structure there, and that allows it to be able to be compacted more to where it stores lots of information, as you can see. You can see the sugar deoxyribose there, the nitrogen-containing bases. The phosphate group there is figure 24.3 is the molecular structure there. And then down at the bottom, you see the different nitrogen containing bases. And the nitrogen bases, as you can see, you've got like 2 on each rung, and they go together. So thymine, adenine, cytosine, and guanine. All right, that's what those go together. TA, thymine, and adenine, I think. Teachers assistant. All right, and then CG, cytosine, and guanine, those go together. So those are basically the base pairs. So this is the double helix structure of DNA on page 489. All right, look on page 490. And like I said, on the top of page 490, you want to know those 4 base pairs. All right, adenine, cytosine, thymine, guanine. You need to know those, and then know that guanine and cytosine go together, and adenine and thymine go together. All right, they only one type of base will fit with the other. The structure of an RNA molecule is similar to DNA, except RNA only just has 1 strand instead of 2, with that double rung there. So it doesn't have the double helix structure. It's just like 1 rung. Look over on page 490 at the bottom, and you'll see the difference between the structures and the difference between them in general. You will find the answer to number 6 on 24.1 in that. That's where you'll find that, in that table 24.1. All right, so that's basically the structure of DNA on 24.1. Have a good day.